In a universe of absolute biological chaos, it is honestly mind-blowing how life manages to organize itself. Picture this. Out of a pitch-black cosmic void, a massive, rotating holographic tree of life emerges. Its roots pulse with this faint crimson light, branching out into a complex web of millions of ethereal, glowing nodes. Welcome to our explainer on patterns in life. Today, we're diving deep into the digital matrix of Earth's biodiversity to see exactly how scientists decode this wildly complex network.
Now think about this for a second. Imagine you're standing in a massive, sprawling library where millions of books are just randomly dumped on the floor. No shelves, no categories, zero order. If I asked you to find one specific book, it'd be an absolute nightmare, right? Well, that's exactly what studying the millions of species on Earth would be like without a rigorous classification system. It would be literally impossible to navigate.
And that brings us to biodiversity. This glowing web of life we're looking at isn't just some random collection of data points. Every single node has a critical purpose. We're talking about everything from microscopic algae in the deep oceans that pump out the oxygen we breathe to the subterranean fungi breaking down waste in the soil. This immense variety is literally what keeps nature stable and functioning. Without this intricate interconnection, life on Earth simply couldn't survive.
Section 1. Building the hierarchy.
To actually make sense of all this, scientists have to play detective. They use 7 strict criteria, looking at fundamental things like cell structure and genetic similarity to sort organisms into a highly structured framework. Think of it like a glowing, descending holographic pyramid. We start at the very top with massive, broad categories called kingdoms. Then, step by step, we filter down through phylum, class, order, family, genus, until we finally pinpoint the exact species. It's basically a biological GPS, tracking an organism from a giant global group down to its exact unique identity.
Section 2. The Evolutionary Timeline.
But you have to remember, this massive pyramid of diversity didn't just pop up overnight. These designs were forged over eons, in the dark, murky waters of evolutionary time. As the environment shifted, the organisms that adapted survived. Primitive single-celled silhouettes slowly morphed and branched out. Simple multicellular forms built up tissue organization, which eventually engineered the highly complex, multi-layered organ systems we see today. Evolution is incredibly efficient. It keeps the old designs that work and constantly engineers new ones.
Section 3. The 5-Kingdom Matrix.
We can map this entire evolutionary timeline using Whittaker's matrix, sorting all of life into 5 distinct, isolated subterranean chambers based on their fundamental traits. Let's step into the basement level, Kingdom Monera. In this pitch-black space, you'll find floating neon-blue prokaryotic bacteria. They don't have a well-defined nucleus at all. Their DNA just drifts around like glowing cyan strands of unorganized nucleoids. It is life at its absolute most primitive.
Moving up a level in complexity, we hit a fascinating contrast. On one side, under this ominous ultraviolet glow, we have Kingdom Fungi. These are heterotrophs with chitin cell walls, acting as nature's ultimate decomposers by consuming dead matter. On the exact opposite side is Kingdom Plantae. Picture a Gothic greenhouse filled with autotrophic producers rocking rigid green cellulose cell walls that actually synthesize their own food. That chemical difference, chitin versus cellulose, is an absolutely crucial evolutionary split.
Inside this dark greenhouse of plant evolution, the leaps in engineering are wild. We start with Thallophyta, like algae, which barely have a differentiated body. Then bryophytes, the mosses, which act as the amphibians of the plant world. But the real game changer was Cheretophyta, ferns that actually developed internal vascular plumbing, namely xylem and phloem. Finally, we reach the towering gymnosperms, like pines, which evolved naked seeds on cones just so they could survive brutally freezing climates.
Meanwhile, in a completely different, high-tech, metallic arena, Kingdom Animalia was taking shape. These are multicellular, heterotrophic organisms that made a massive evolutionary trade-off. They completely ditched rigid cell walls. And what did they get in return? The ability to move, hunt, consume, and most importantly, rapidly react to their environment when danger strikes.
Animal blueprints upgraded drastically over time. You go from the basic cellular pores of a sponge in phylum Porifera to the primitive, single-opening tissue system of a hydra. But then, nematodes, or roundworms, engineered a highly advanced 2-opening digestive tract. And that eventually led to chordata, organisms that developed an internal notochord, which laid the foundational architecture for our own backbones.
Now, why does any of this matter to you and me?
Because this biodiversity isn't just a concept in a textbook, it is a literal physical shield protecting human society. During the 1999 Orissa supercyclone, dense mangrove forests acted as a biological wall that significantly reduced human destruction. High biodiversity acts as a barrier against zoonotic diseases like monkey fever. We even rely on soil microorganisms to chemically break down toxic environmental pollutants. This web of life is literally keeping us safe.
Section 4. Scientific Identity and Nomenclature.
To study and protect this vast network globally, we had to eliminate the absolute chaos of local languages. Imagine this: local names like tiger, baj, or tigre burning away into ash, leaving behind a brilliantly glowing cyan code. That code is Carolus Linnaeus's universal binomial nomenclature system. By assigning the tiger the scientific name Panthera tigris, a scientist in Tokyo and a scientist in New York know exactly what species they are talking about. Zero confusion.
To make this system completely flawless, it runs on 3 strict rules. First, the name is always a 2-part Latin tag, genus and species. Second, the genus always starts with a capital letter, while the species is entirely lowercase. And third, the formatting is non-negotiable. It must be italicized when printed or underlined when written by hand. It's a perfectly engineered global ID tag for every single organism.
And precise classification is no joke. It has literally saved species from being wiped from the history books. Take India's endemic Sangai deer, found only on the floating Phumdis of Manipur. In 1951, it was officially declared extinct, gone forever. But just 2 years later, thanks to precise classification techniques, scientists were able to miraculously rediscover and correctly identify a surviving population. Though, to be fair, they are still highly endangered on the IUCN Red List today.
Of course, as our technology evolves, our classification matrix has to evolve with it. In 1977, as we finally developed the tools to read DNA directly, scientist Carl Woese gave our entire system a massive upgrade. He looked past physical traits, diving straight into the genetic code, and reorganized all life into 3 overarching domains: bacteria, archaea, and eukarya.
So, as we rapidly zoom back out from this binomial matrix, passing the 5 kingdoms, and watch this massive tree of life fold back into a single perfect data point in the dark void, it leaves us with one incredibly provocative question. Evolution never stops. If our environment were to completely change tomorrow, what bizarre new structural adaptations will evolution engineer next? Definitely something to think about.